Artificial intelligence in drug discovery is a relatively new field. It's a very important field. And today we're speaking with one of the most prominent voices in AI and drug discovery. I'm Michael Krigsman. I'm an industry analyst. Thank you so much for watching. Before we begin, please subscribe on YouTube and subscribe to our newsletter. You can do that right now. Alex Zhavoronkov, he is the CEO of Insilico. Tell us briefly about Insilico Medicine and tell us the things that you're working on.
We are focused primarily on applying next-gen AI techniques to drug discovery, biomarker development, and also aging research. And we focus specifically on two machine learning techniques. It's generative adversarial networks and reinforcement learning. So those techniques we are most expert in our field, and we use those techniques for kind of two purposes. One is identifying biological targets and constructing biomarkers from multiple data types, and also generating new molecules, new molecular structures with specific set of properties. So we were one of the first companies, possibly the first one, to generate new molecules using this new technique called generative adversarial networks. It's kind of AI imagination, and validate those molecules experimentally.
Give us some context. What is the drug development pipeline? Why is it so hard? Let's talk about that, and then we can shift to how AI makes that better, makes it easier.
Drug discovery and drug development is a very lengthy process. And it's also one of those processes where you've got more failures than successes, actually much more failures than successes. It takes more than $2.6 billion to develop a drug and bring it to the market to address a specific disease. And that's after the molecule has been tested in animals. And also there is 92% failure rate after the molecule has been tested in animals. When it goes into humans, it fails 92% of the time. So the process is not only lengthy, but also risky. Usually the time it takes to discover and develop a molecule is around a decade. So people who initiate the process are not always there when the molecule launches. And the process is comprised of several steps. So first one is hypothesis generation. So you come up with a hypothesis, a theory of a certain disease, and identify relevant targets. So you theorize about what kind of proteins are implicated in a disease condition and what proteins are causal. Afterward, you go and develop either an antibody or a small molecule for this protein target. And if you're developing a small molecule, I usually start with screening large libraries of compounds that might hit this particular target and do all kinds of experiments to see how well those small molecules bind to this target. Afterward, you select several hits. You identify what kind of molecules fit best for this protein target, and start doing all kinds of experiments on those molecules to see if they work very well in a biological system, in a disease-relevant assay, in a mouse, in a dog or other animal, and then you file for IND with the FDA. To get the molecule into clinical trials. So after that process is complete, we are getting into drug development and starting clinical trials. It starts with phase 1, which is safety. Phase 2, you test for efficacy. And phase 3, you test for both in a larger clinical setting, a larger population. And then you might want to go for a phase 4 or start launching the product. Mm-hmm. And post-marketing research. So that process takes more than 10 years usually, and fails 92% of the time. So with AI, you can really play in pretty much every segment, from early stage drug discovery, where AI can assist you with a hypothesis model and essentially pulling out the needles from the haystack with a target ID, with small molecule identification, with virtual screening, with generation of novel molecules with specific properties, with planning your clinical trial, clinical trial design, with enrollment of the clinical trial, and then also for predicting the outcomes of clinical trials.
Where does AI begin to shorten that process, make that process better?
So if you go at the very early steps of the pipeline and start working on the hypothesis generation and target identification, usually you have multiple kind of paths to pursue. So one path is to look at the literature and identify promising areas that have been uncovered by scientists in the past and were published in peer-reviewed literature. And ideally, these targets, those hypotheses, were not implicated in this disease that you are looking at by somebody else. So AI can help you mine massive amounts of literature and also other associated data types to identify signals that a certain target might be implicated in the disease. So we at Insilico usually start with grants data. So we look at biomedical grants. We monitor about $1.7 trillion worth of grant money over the past 25 years. And then we look at how those grants progress into publications, into patents, into clinical trials, and then into products on the market. So we follow this idea from idea to, and from money to money. So from money on the market. And we also look at how money becomes data. So usually when the government is supporting certain study, the data needs to be reposited in a public repository for other people to replicate it and also for common good. So we try to follow the money into data. If the data is not there, we try to contact the scientist and get the data from the scientist, or to encourage the scientist to put the data into the public repository. So we start with text databases, but also link this data to to omics data. It's basically everything that ends with omics is called omics data. So transcriptomics, genomics, metabolomics, you name it. So metagenomics. And we work primarily with gene expression data. So we look at how the level of expression of certain genes or entire networks changes from, let's say, healthy state to disease. And we deconvolute those changes, those signatures of disease into individual targets, establish causality models, and identify what kind of proteins could be targeted with a small molecule. And then we go back into the prior art, into text, and see if anybody has published anything that strengthens our hypothesis. It doesn't necessarily mean that our hypothesis is wrong if the signal is not there in text, because sometimes the humans just couldn't really associate a certain target with a disease using older methods. But it gives us a little bit more confidence to see that somebody already touched on this challenge and on this target before.
So, Alex, is the key then at this point that the various AI techniques that you're using enable you to discern patterns in the data that otherwise would not be, those signals, as you said, that otherwise you could not pick out? Is that the key issue here?
So yes, but really we are aggregating enormous amounts of data that is just not possible to process using human intelligence. And we're also aggregating and grooming those data types together. And sometimes those data types are completely incompatible, and it's impossible to, just suture them together using standard tools. So you really need to train deep neural networks on several data types at the same time in order for them to generalize and in order for us to be able to extract relevant features that are present in several data types at the same time. So some of the data types that we work with are completely incomprehensible to the human mind, to the human intelligence, like, for example, gene expression or movement or cardiovascular activity scanning or ultrasound, for example. So we manage to bring those data types together using AI and then identify relevant targets that basically trigger a certain condition.
At Insilico, is your core competence in the biology and medicine or in developing the AI techniques? And is it possible to even split those two?
So in our case, we are good at both. And we hire competitively internationally. So we actually hire through competitions where we put very challenging tests out in order for people to try and solve them very, very quickly. And those challenges are usually in combination of, developing an AI method plus solving a complex biological or chemical problem. However, when you're looking at really great AI scientists, they're usually not great in biology or great in chemistry. They're good in math. And that is why some percentage of our company are just great mathematicians who are developing novel methods for bridging chemistry and biology using deep learning, for example. And part of the company is specifically focused on applications of already existing techniques like GANs and reinforcement learning to existing problems in chemistry and biology. So those people are usually on the applied side, and they know both chemistry and biology, and they can talk to the mathematicians, and they can do some basic research in AI as well. And of course, we just have pure play biologists and chemists who are also necessary in order to validate some of those some of the results of our AI. So that's why we have such a large and diverse and international team, because you really need to have those three areas covered: the methods, the applications, and validation.
We have an interesting question from Chris Peterson on Twitter, who says this. He says, grid-based parallel Fortran programs are still being used for some pharmacokinetic and pharmacodynamic studies. Do you see AI replacing the old school code, enhancing it, or advancing in parallel?
So I think currently we need to advance in parallel. And of course, some of the old techniques and some of the very primitive mold dynamics are still being used by really top experts in drug discovery today. But most of those methods are being significantly accelerated by high-performance computing and AI. So typical software that's been around for a very long time, like Schrödinger, for example. The company has been around since '92. These guys made major breakthroughs in multiple areas and kind of managed to advance older algorithms to solve very complex problems. And I think that, at Insilico, we try to reinvent everything from scratch, and we write our own software. But of course, we know many of our collaborators who just like to take small pieces of our kind of big salami that we're developing and play around with it today. They might be using some more classical tools that you cannot get around around today. But
Why?
ideally, you need to have a seamless pipeline which identifies the targets, generates the molecules, and runs those molecules through a large number of simulations in one seamless pipeline. That's what we are building, and that's our holy grail. But of course, many companies, many groups are trying to do the Lego game and try to use multiple tools with varying outputs to solve the same problem.
Why do you develop your own tools?
Again, just because many of the methods that we're using are so new that they're incompatible with older tools. And there are many groups that claim to do AI, but essentially what they're doing is they're mechanics shops taking off-the-shelf software and trying to bridge some gaps in pharma R&D using those tools. We don't do that. We develop everything from scratch. So from target ID to small molecule generation.
So now we have spoken about using your techniques to uncover potential candidates. The next step is evaluating. So first we have to uncover possibilities, and you do that by aggregating all of this data and then mining that data using the various techniques. Now you've done that, how do you evaluate the candidates that you've uncovered initially?
So usually when you are left with a list of protein targets for a specific disease, and you are trying to prioritize, you try to annotate those proteins with as many scores as possible. So you're looking at whether this protein target has ever been implicated in toxicity. How is it connected with everything else? Which tissue does it play in more? How does it interact with other proteins? Is it druggable? Is it druggable with a small molecule or with an antibody? Did anybody else touch it? What is the patent space around the molecule? Has anybody tried taking it into the clinic with a small molecule or an antibody for a specific disease? So there are many scoring functions that you need to consider. And at the end, when you basically are left with a very small set of targets, then you also test them in a variety of biological systems to see which one is more relevant for your disease of interest. And I'll give you an example, a case study. So, for example, we are very interested in fibrosis, and fibrosis is not a very simple process to describe, and there are multiple types of fibrosis. So there is IPF, so pulmonary fibrosis. There is smoking-induced fibrosis in the lung. There is aging-induced fibrosis in the lung. So we've identified more than 120 types of fibrosis by comparing normal tissue to tissue afflicted by a certain condition that is associated with fibrosis. And we just recently did a case study where we looked at IPF, so pulmonary fibrosis, identified a list of targets for this condition, and our list was 50 targets. And we looked at when those targets are more active and more disease relevant, at what stage of the disease, because I think if you kind of catch it later or address it later when there is just so many symptoms, you are going to be treating the symptoms, not the cause. And in our case, we've identified a large list of targets that are likely to be very relevant early in the disease progression. And then we looked at what targets are novel. So you look for novelty. So what targets people did not focus on as much. So you don't want to focus on old targets. Then we looked at what targets are druggable, so where we could actually come up with a small molecule from a vendor library, or we can generate a molecule from scratch. And then we looked at what targets could be validated in a specific set of assays for fibrosis.
And where's the impact of the AI techniques that you're using in this?
So usually it's for scoring. So you identify multiple scores for those targets. So in our case, the target is annotated with more than 50 scores. So whether it has been implicated in a certain condition before, whether it interacts with other proteins in a specific way, whether it is likely to lead to toxicity. So those predictors that basically give you this kind of score and a probability that this target is the most relevant one, these scores are deep learned. So they come from we developed them using machine learning.
We have another interesting question from Twitter. This is from Shreya Amin, and she says, how does this type of research that you've been describing using AI in the process compare between academia and industry?
Sure, it's a very, very good question. So in the industry, in big pharma, people are a little bit less adventurous. They are trying to develop the various techniques to really solve a problem and make incremental changes. It's not for publication purposes. In academia, people are much more innovative and adventurous, and of course, they try to publish. And that's where the innovation comes from, primarily. So we at Insilico, we sit in between academia and industry. So we publish at the rate of about 2 research papers a month. That is a lot for even some of the academic groups. Just to also prove the concept and explain where we're going. So academics, I think, are much more productive nowadays when it comes to developing new methods and showing new directions. However, the disconnect between really good computer scientists that are developing novel techniques that might be relevant for drug discovery, they very often are so far away from biology and chemistry that they put the papers out, and the paper is really, it's brilliant from the machine learning perspective, but it's really, really poor from real world applications. And very often they don't really understand that they overfitted somewhere, or if it's a completely irrelevant output that they're getting or input, only after somebody tries it in biology and chemistry. So very often, and nowadays it's actually more prevalent, a lot of people put papers on archive, so in a repository, and with a catchy title, so it goes viral and gets picked up by the browsers or by Google or by some news outlets, and they get recognition and PR for this work, but then you try to replicate what they did or even just read the paper carefully, and you realize that it's not going to work in the real world. And I think those kind of papers and those kind of efforts, early efforts by academic groups, and specifically without going through peer review, also put a lot of skepticism in big pharma. So people just don't think that many techniques are relevant or applicable or transformative for their business.
Let's talk about the team construction aspect, because one of the things that you've mentioned a couple of times is the importance of both the machine learning capabilities as well as the biology capabilities. And these are very specialized skills. And so how do you construct teams that enable both sides to work together and create something that one or the other could not do alone?
So that's another very good question. And in our case, that's one of the reasons why we are growing so slowly. So we've been in business for 5 years now, but we are still 66 people. And one of the reasons why for the slow organic growth is because it takes time to really integrate the AI scientists with biologists and chemists. So it's very difficult to find people who are good at both at the same time. So usually you are good at math, or you're good at you're good in chemistry, or you really need to have some good programming skills to be able to do an API. And properly combine your technology with somebody else's. So we try to work in teams of 3 or 4 on specific therapeutic projects, where one person is very good in chemistry or biology, one person is good in AI, and another person is good in just basic IT. So it's basically teams of 3 or 4 people. And on top of them, there is an infrastructure, an organizational infrastructure that helps manage those teams. And we also separated the kind of pure play AI team from everybody else's, so they could work on the methods without being brought into the applied domain. So getting this kind of talent who are willing to really contribute to methods development and develop novel algorithms, that is very, very difficult. Getting people who are good in application of already developed methods, that is rather easy. Getting the two to work together, that is very hard. And to do this, we try to pursue organic growth and work on projects in small teams.
And in fact, we have a question from Twitter on this subject of your business model. Chris Peterson's asking great questions. Thanks so much, Chris. Is asking are you contracted to look for specific therapies, or are you developing molecules from scratch and hoping to license them for clinical trials through distribution?
So we've been in business for 5 years, and we've explored multiple business models. As an AI company, you have to explore because otherwise it's very, very difficult to scale on one business model, and it's also quite risky. So we started as a service company and started partnering with pharmaceutical companies and with biotechnology companies, and also with venture funds, where we provided a service or provided a system to them. So we learned the applications that people are looking for and started developing our own small molecules, discovering our own small molecules and then licensing them. So our current business model is actually very simple and actually allows us to scale. So we work with venture capital firms that really know the business of biotechnology and are pursuing drug development and drug discovery. So they guide us into where we need to identify targets and generate small molecules. And then they form teams around those small molecules and targets, fund them, and let them do a little bit more validation and development of those target and molecule associations. And what we get a small upfront payment initially, and then we get milestone payments as the molecules progress through the various steps of validation. And then we get some royalties. So usually if you consider the biobucks or the future revenues that might come from the molecule, those deals are very, very substantial. but initial payment is rather small. That is why we have another business that is software licensing business, where we license some of our software tools to others to generate some revenue and ensure that we are sustainable, consistent, and also get some feedback on how well the software works.
Okay.
If we need to add more features. And another business model is that we do have some joint ventures. For example, a joint venture with a company called Juvenescence, and they are developing the molecules that we provide to them.
Okay, so you have a diverse range of things that you're working on and trying that support your business model efforts, essentially.
Correct, but what we're mostly interested in is not the immediate revenue. In most of those licensing arrangements and engagements, we get some data back, and we pretty much became one of the largest data factories in the world, getting data back from preclinical experiments.
That's interesting. We have another question from Twitter, and this is from Trovato Christian. He is a biomedical engineer, and he's a PhD student in computational biology in the Department of Computer Science at Oxford. And by the way, I find it very interesting that computational biology falls under the Department of Computer Science rather than the Department of Biology. So his question is, are there any examples of drugs developed by AI only?
At this point of time, there is no such example. You always have a human in between. I hope that in the very near future, we'll be able to show that the pipeline where no human was involved, from target identification to small molecule generation, might be able to churn some of those molecules, some of those promising molecules. But at this point in time, the experiment is king. So unless you can validate your techniques experimentally, it won't really go forward. So I have never seen any example of a molecule ending up even in mice at this point in time that is completely generated using AI.
What's the obstacle preventing using AI to go from beginning to end?
Well, because of the failure rates in pharma in general. So there are very, very few success stories to train on. And those success stories are very, very diverse. So in some areas, it's easy to validate whether your algorithm is producing some meaningful output. But in many cases, you really need to go and validate at every step of the way. So that is why when you're building this salami that is allowing you to go end to end, you need to ensure that you validate every slice of the salami and validate it internally, but also validate it with external partners. That's what we are trying to do as well.
So eventually that data may be there, but it sounds like it's just far too early at this stage.
At this stage, nobody tried to virtualize drug discovery completely using AI and do it seamless without human intervention. In many areas, it's actually not possible just because biology is so diverse and medicine is so diverse that it's very, very difficult to have a solution that would fit all. So that's why people are going primarily after cancer, just because it's a little bit easier to validate. And specific types of cancer, like for example, solid tumors, where you can do a xenograft and see if the tumor shrinks in a mouse if you feed it, if you give it a specific molecule. So there needs to be validation at every step of the way. And at this point in time, those end-to-end pipelines will work only in certain therapeutic modalities.
Let me ask you another question from Twitter, and this is from Shreya Amin. Again, a great question, interesting one. And she says this. She says, using existing AI techniques, which areas, from the perspective of types of drugs, diseases, conditions, and so forth, are closest to breakthroughs or have made the most progress, and what's most difficult?
I'll give you an example that I'm very, very familiar with. So we've got some JAK inhibitors, so Janus kinase inhibitors that are developed completely using generative adversarial networks and reinforcement learning. So I think those are kind of the most promising techniques for de novo molecular design, period. We're currently in mice with those. So went all the way from enzymatic assays to mice, and showed that we can now achieve selectivity, specificity with those molecules, and those molecules have many other properties. And those are pretty kind of common techniques nowadays, both the GAN that we used and the reinforcement learning technique that we used. It's not something super new. So we actually switched our R&D into a slightly different direction.
Where is all of this going over the next, I don't know, 3-4 years, 2 to 4 years? Let's not go out 10 years, but over the next few years, where is this going to be?
I think that companies like ours are going to put much more emphasis on their internal R&D instead of collaborating with big pharma, because collaborating with big pharma is usually a path to nowhere, because it's either death by pilot or it's they just ingest this expertise internally and catch up. But at the same time, they are so bureaucratic that it's very difficult to change. And at the same time, at the CEO level, big pharma companies are more focused on increasing sales or buying other companies to increase sales or to get late stage clinical assets. So phase 2, phase 3 assets. So the internal R&D is actually not being viewed as a huge priority. I mean, regardless of what they think, it's fact. And usually it's kind of the 15-20% on the income statement that needs to be there, because otherwise the investors are not going to invest in the company. But the productivity of this internal R&D is usually very low. So I think that smaller biotechnology companies that embrace AI and embrace virtualization of drug discovery, they are going to be very successful. And there are several cases that I admire in the industry, like, for example, Nimbus Therapeutics. This guy has managed to virtualize the entire drug discovery and development process and get some, phase 2 assets to market. And license them. So as AI improves and starts solving more problems in the pharmaceutical R&D pipeline, so from hypothesis generation and target ID and small molecule generation and prediction of the various properties of the molecule in clinical trials and better stratification techniques, I think that people who really understand the process and can virtualize it will be the winners. And so far, I know several companies that are doing this. So some companies are working with us, some are in the stealth mode, and I think they are going to be the winners going forward. But when you talk about drug discovery, 2 to 3 years, it's actually a very, very short time. In many other areas of human development, if you ask me to plan 5 years ahead, I won't be able to, because things are changing very quickly. In pharma, that's not the case. We really need to do the experiments and get things right.
Do you want to just very briefly tell us about the last research you did on either longevity or smoking? I know we're out of time, but just very briefly.
Sure. We just published a very fun paper showing that smoking accelerates aging. One of the areas that we are focusing on is age prediction using multiple data types. So from pictures, blood tests, transcriptomic data, proteomic data, microbiomic data. We use this data to predict the person's age reasonably accurately. And we then look at what kind of interventions or behavioral modifications, what kind of lifestyles contribute to that person looking younger or older. And we did this exercise in Canada. We worked with the University of Lethbridge and the government of Alberta to process a large data set of smokers and non-smokers of varying ages, looking only at anonymized blood tests, just very, very few parameters from a recent blood test. So first of all, we built a predictor of the smoking status. So now I can, with reasonable confidence, say whether you're smoking or not, looking at the blood test. But also we showed that people who smoke, they look older to the deep neural net trained on their blood tests than non-smokers. And once we published, it actually went rather viral, and we got very positive feedback. Like, for example, my daughter is considering quitting smoking just because she doesn't want to look old. So people don't really care about their health, but they really care about how they look. So if you don't want to look old, just quit smoking.
Okay, great advice. Alex, thank you so much for taking time. Everybody, please subscribe on YouTube. Check out CXOTalk.com for lots of videos and subscribe to our newsletter. Have a great day, everybody. Take care. Bye-bye.